My name is Angelika Manhart, and I am a lecturer in applied mathematics at the mathematics department of UCL, and I am associated to the Institute for the Physics of Living Systems. What my group does is we use mathematical simulation, analysis, and modeling to understand cell biological systems. Let me give you some examples. The two common themes in my group's research is movement and scales. So movement can mean the movement inside of cells, of single cells, but also of cell collectives. In the first example, movement inside cells, I'm showing you the movement of nuclei in muscle cells. So a muscle cell contains several nuclei. That's the lower picture that you see here. And those nuclei need to be positioned in a way that keeps the muscle cell healthy. For the movement inside of cells, main questions are: how are forces generated that allow single cells to move? How do they turn? How do they change shape? And so on. So you see in the lower picture, you see the picture of a keratocyte. That's a fish cell, and it has a network structure that allows it to move. And this network consists of actin filaments that interact with many regulators. And in the third example, the movement of collective cells, you see in the lower picture, you see bacterial colonies. So these are called myxobacteria. They live in the earth, and they form wave-like patterns. Now, with regards to scale, all of these effects that we're seeing, correct positioning of nuclei, single cell movement, collective dynamics, the reasons for what we are seeing are usually on a much finer level of detail. So that can be individual cells interacting for collective movement, networks being created and broken apart when they interact with their regulators, or nuclei where fibers emanate from their membranes interacting with each other. And so I want to find the right mathematical tools or pick the right scale to understand this phenomena and connect the different scales so that we are able to understand macroscopic effects, that we are able to understand collective dynamic of bacterial cells, of cancer cells, how single cells move, how they get to the right place, how they sense their surrounding and change their shape, or how nuclei are positioned correctly in muscle cells, and how that affects muscle function in that case. So the general cycle that my group goes through is we work with experimental collaborators. So typically, we don't do the experiments ourselves, but we work with the data, and the data then informs a hypothesis, and that informs what is the best model we can use to test this hypothesis computationally. Sometimes these models can be analyzed with pen and paper, but typically they need to be solved numerically in order to understand their behavior. And then we reach certain model predictions, and they can be tested against the data. The main modeling tool that I use are differential equations, which come in a number of different flavors, and I use whatever flavor, so partial, ordinary, stochastic differential equation, whatever is necessary and is the best tool in order to answer the biological question at hand. Let me show you 3 videos to give you an idea of the kind of simulations or models that we use. So in this first example, this is related to the nuclear positioning in muscle cells, and the red circles represent nuclei. I'm showing you 2 videos in parallel, and they are based on very different models. So the left one is based on a much detailed model where you will see individual microtubule filaments appearing, interacting with each other, shrinking, and so on. Whereas on the right-hand side, I just have a coarse-grained model where there's a force field around each nucleus. And the coarse-grained model has been informed by the agent-based model. And what you can see when we watch the video is that they behave incredibly similar. So you could almost not tell the difference if it wasn't for a different visualization. So we see how the microstructure leads to an averaged macroscopic behavior that also helps us understand the main features of the positioning. In that case, that you have typical spacing between nuclei and typical patterns that can be used, for example, to diagnose certain muscle diseases. In the second example, I'm showing you work where we try to understand what determines the equilibrium length of actin networks, which is then connected to, for example, the size of cells. And you can see on the right, you see data from our collaborators. On the left, you can see simulations of a stochastic network model. So that is actually not differential equations based. And you can see that we can predict a certain equilibrium length that is reached and general features of the data match with the simulation. And the third example is to do with collective dynamics. So in the first video, you see an experimental video. So you see density waves of a colony of these myxobacteria. And we use two different modeling approaches which were informed by each other. The first one is an agent-based model. So if you ignore the colors, you can see that we have similar interacting wave patterns forming as in the experimental results. And then this model, which is very difficult to analyze to get insights out of, we used it to derive a PDE-based model where we see the same features, so we have interacting wave patterns, but it's easier to understand and get insight from and make predictions for how, example, the behavior will change if you have mutant cells where certain processes are defect. I hope this gives you an idea about the kind of problems and the kind of methods that my group uses and why they are important. I want to show you, give you a quick idea of the kind of people that I typically work and publish with. So here are some names, but you can find more details on the webpage. Feel free to get in touch if you want to talk about a certain project or you have some questions.